Hello everyone. Today I am going to start the next part of permanent tissue, as I have started parenchyma and completed parenchyma in last class. So there are two other permanent tissues are there. These are collenchyma and sclerenchyma. So today I am going to discuss this one. But before starting this, I want to say, so if you like my class, please give like, okay, and please share my share the video, and how much you share, this much I can reach the students. Okay, I want to help you, so I need your help also. Please subscribe my channel and don't stop video in middle. Please try to watch till the last. You'll get some information, and you can get some notes also from here. You'll get a clear idea of the chapter. As I told you, that tissue is not a very interesting like self. Maximum people don't get interest here. So I'm trying to making it interesting. So follow my class till the last. Okay? So let's start discussion about collenchyma today. So collenchyma is a tissue which helps to give flexibility, provides flexibility. Flexibility means, you have seen the climbers plants. In case of climbers plants, you can see the climbers can bend, and the tendrils present here which can climb by support, means the thread-like structure. Okay, these are spring-like actually sometimes. So these can bend in any side. This is possible for the collenchyma. Okay, now you got in climbers plants, mainly we are getting this one. Okay, the climbers, part of the stems, okay, and tendrils, okay. Now, where is the location? In which part we are getting? In my class of parenchyma, I told you parenchyma is present just below the beneath the what? Epidermis, the outer covering of plants. Okay. So collenchyma is also present just below the what? Epidermis, but it is just directly related with epidermis. It is present here, directly related. It is epidermis. Okay, and here this one, these layers are what? Collenchyma. Okay, these are the collenchyma, and after that parenchyma is present. Now what happened? Location we have known that present below, or you can say beneath the epidermis. Now epidermis of which plant? Epidermis of dicot stems, or you can say the stems, or and like the leaf. Okay, you can see the leaf stalk. Now what is leaf stalks and what is dicot? Okay, as I told you, dicot means dicotyledonous plants. So di means two and cot means cotyledons. Two cotyledons are present. So what is cotyledons? Cotyledons means the embryonic leaf coming out. Okay, there are two types of cotyledons, monocot and dicot. Here we are getting in dicot. Which one is present? Collenchyma is present. Okay, so dicot types of plants in mango plants. Okay, so you can see where you can see the seeds. You can differentiate it by seeds, and you can differentiate it by leaves. In case of monocot, you can see the single leaves are coming out, like a coconut plant, sugar cane, then a banana. Okay, but in monocot plant, dicot plants like mango, jackfruit, you can see what happened. The leaves are, so many leaves will come out, or at least more than one leaves will come out from the soil. Okay, from the embryo. And the seed, if you open the seed of mango, you can see the two parts are here. Okay, two parts are joined separately. Clearly you can see. But in case of nut, in case of corn, in case of coconut, you will get a single cell is there. So like this we can differentiate monocot and dicot. Di means two cotyledons. Okay. So means two leaves, embryonic leaves will coming out from this one. Okay. So in case of dicot plants, you will get, like in case of mango, jackfruit, you are getting these types of Collenchyma. Okay, beneath the epidermis. Now, what is the structure of this one? There is a figure outline is there. I have drawn for you. This one is the structure of Collenchyma. How it is now? Collenchyma structure. See how it is looking like. Here you can see the cells are living. Okay. So living cells are present here. Okay. So all the cells contains protoplasms, cytoplasms and nucleus. Okay. You can see here what happened. Distinct nucleus. These are the nucleus here, you can see. Nucleus. Okay. Here you can see this part, cytoplasm is present here. Okay. Cytoplasm present here. So here also abundant cytoplasm present here. Okay. So cytoplasm, as it is a living cell, so cytoplasm is present here. Okay, now number 4. What happened here? You see a big vacuoles. Big vacuoles present here in the cell, you can see. The most important thing is the cell wall. Cell walls are very thin, you can see from the picture. The walls are very thin, but important is the corners. Corners are what happened? Corners are actually very thick. Corners are thick. Okay. Thick with the presence of cellulose. Okay. With cellulose is present here. Cellulose and pectin. Cellulose you already know, a complex carbohydrate. Pectin is also a complex carbohydrate. It is also a hard a starch. Okay, a complex starch. This one. So there are cellulose and pectin present in the corners. You can say how hard these corners are. Okay, so the borders are, you can see, hard. Okay, is this clear? These parts are made by cellulose and pectin. Next one is there. You can see that the cells are actually elongated shape. That's why already I told you why it is elongated. The structure is not equal in all the side, like parenchyma. The cells are very thin, but their corners are very thick. So the shape is elongated. Okay. Next it is that you can see the intercellular spaces are present here. So intercellular spaces are absent. Intercellular spaces are absent here. You can see from the picture there are no intercellular spaces are there, as the corners are blocked completely with cellulose and pectins. So they are tightly joined, so there are no intercellular spaces like parenchyma. What we get in parenchyma, there are holes are there. Okay, they are in between the two cells, that there is no intercellular spaces between two cells. Okay, this much is clear. So see, you can see these parts are already covered. There is no intercellular spaces. So this is the structure. But one more point we want to discuss, that sometimes they contains what? They contain chlorophyll. So if they contains chlorophylls, okay, so it means in their functions we are getting that they are able to do photosynthesis. Okay, able to do photosynthesis. So they produce sugar, means their food, glucose. So due to photosynthesis, they can produce sugar. Okay. Next part, they provide mechanical support to the plant. Mechanical support to the plant. Okay. This is the function of Collenchyma. So I think this part is clear. Okay. Now, from this picture you are getting this point. So the point I have given, you can take as a note. Now let's start discussion about sclerenchyma, another permanent tissue. Okay. Sclerenchyma is a supporting tissue of the plant. Okay. It is present in all the hard part of the plants. It is present in root also, stem also, in the veins of the leaves also. In the veins of the leaves. If you see that in the leaf, in the veins of the leaves it is present. Okay, these are called the veins, and in the vein sclerenchyma are present. Okay, so in all the hard parts, it is also present in the seeds cover, like seeds covers of nuts, coconut shell. There sclerenchyma are present. Now we are going to discuss about the location where it is present. Present in all the hard parts of plant. Okay, like even the seed covers, roots, stems. Okay, so in these parts it is present. Now, according to structures, sclerenchyma are of two types. You can see one, you can see like fibers. Fibers like structures are there. One is sclereids. So one is fiber, one is sclereids types are there. So for that we have to know the structure of sclerenchyma. So let's discuss about structure of sclerenchyma, how it's looking like. So the most important point of structure, cells are dead here without protoplasm. So there is no protoplasm present here. Okay, this is very important point. Next you can see the cell walls. For that I am going to draw a structure. So the border, if you see, they are very much thick borders are present here. Okay, the borders are very thick. Thick borders are there. So the cells are dead. There is no what? No protoplasm present here. This is the dead permanent tissue. So this is the transverse section. If you cut in transverse section, you will get this structure here. Okay. So here we got the cells are dead here. No protoplasm is present. And the cell walls are very thin. Sorry, very thick. And it's made up of, cells wall actually made up of here with lignin. Okay, lignin is present here. Now what is lignin? It is a chemical structure. Okay, it's organic chemical, and these are actually a polymer. So polymer, as I already told in DNA structure, polymer means it's made up of so many monomers. It's a complex chemical structure. So it is a chemical structure, organic chemical structure made up of this one border. And side by side here, cellulose and hemicellulose also present. Okay, with this lignin in the border, cellulose and hemicellulose also present here. But the main part is why it is so hard, because of the presence of lignin. The structure, you can say, next we are going to the structure, longitudinal structure of the sclerenchyma. How it is looking like. Actually, as I told you, fiber-like structure is present here. So in case of fiber, you are seeing the body is like that. It is a cells are here long and narrow. Okay, narrow, fiber-like. Okay, so here fiber-like. It means little bit like thread, because both the sides are pointed here. So if you see the longitudinal structure, how it is, this part is called lumen. Lumens means an empty space, and these are the cell wall. It is a dead cell, so there is no protoplasm present here. Only the empty space is present in between a fiber. So it is a fiber-like structure. As I told you, there are two parts of sclerenchyma. One is looking like a fiber, another is sclerites. So we got there is no intercellular spaces are there. No intercellular spaces. You can see that how they are joined tightly. So there is no intercellular spaces. But one thing is that cells are joined there with the pits. Okay, so pits means some holes are present between two cells. Okay, cells are joined with the pits. So here cells are joined with pits. Pits means empty space. Okay, so through these empty spaces, cells are joined together. This is a very important point in sclerenchyma structure. So this part is actually about the fiber-like sclerenchyma. Now we are going to discuss what is sclerites. Sclerite structure, you can see in some reduced form of sclerenchyma. Sclerites. What happened here? In the reduced form of sclerenchyma sometimes take different shape. Here, the shapes are not fixed actually, but not fiber-like. You can see spherical, or you can see the oval shapes also present. Oval shapes. Cells are like these. Cells. Okay. They are scattered. Okay. Scattered in plants. They are scattered in different parts of the plants. Cell walls are very thick, and lignified cell walls are present here. Okay, this is the sclerite is a structure is not fiber-like. Here you can see the spherical or oval shapes are there, and what happens? Cell walls are very much thick, and is present and scattered in different parts of the plant. It is present. Suppose if present, you can see the outer, you can see core part of the apple. You can see the outer cover of the guava. Here you can see the body is not very smooth. Okay, very rough cover is. So it's made by sclerites. That's why it's looking like that. Okay, and in middle part of the apple, you can see the hard part. It is also made by sclerite. So sclerites are making the hard. It is very hard part. So if this much is clear, I think the fiber is another structure, and scleroid is present. Scleroid is another structure. Now, what is the function? As we already know that fiber is there, and fiber is actually what to make functions. What are the functions of sclerenchyma? So they also provide mechanical support, or actually you can see these are the supporting tissue of the plant. Okay, they are they form the hard and very rigid structure of plant. Okay, and fiber, what do they do? Fibers are using where we know in textile industries. Textiles. In textile industries, we are getting the fibers are using to make cloths. Okay, the thread in the cloths are made by what? Fibers, and we are the scleroid is actually making this fiber-like structure. Okay, so in jute plant we are getting easily this one. This is the fiber using to make the cloths. One more third point is that scleroids are, as I already told you, making the core part of the scleroids are making hard parts of the fruits, veins. And point number 3, scleroids are present in seeds, hard part of seeds. Okay, hard parts of seeds and bleachers of fruits. As I told you already, guava, the bleachy parts, the outer structure is very rough. So these bleachers are produced by sclerenchyma tissue. So it's already clear what is the difference between collenchyma and sclerenchyma understood. One is living, another one is dead. Okay, how it is working, like function of the sclerenchyma and function of the collenchyma, they are structured. Okay, so today up to this. Thank you. Next class, I start xylem. Okay, thank you.